Hello, everyone. My name is Dan Fu. I'm Assistant Professor of Chemistry at the University of Washington, Seattle. It is my great pleasure to present the final report for my Beckman Young Investigator Award-funded project, Quantitative Single-Cell Phenotypic Imaging of Multicellular Systems. Life begins with the single cell, which divides and differentiates into the embryo and then the human body. In adult life, there are over 37 trillion cells that can be separated into over 200 different cell types. In biology classes, we learned that a cell looks like this, but in the body, cells come in widely varying size, shape, function, as well as the potential for growth, proliferation, and differentiation. These properties are critical to maintain tissue homeostasis. Because cell heterogeneity plays a critical role in tissue function and disease development, such as cancer, it is necessary to analyze single cells in order to fully understand tissue function and dysfunction. Over the past decades, there has been a revolution of single-cell technologies to understand what single cells do and why they do what they do. You have probably heard about these terms a lot already: single-cell genomics, single-cell transcriptomics, single-cell proteomics, and single-cell metabolomics. These single-cell technologies are transforming our understanding of cell biology and disease, but they do not tell us everything about the cell. Just like knowing a car engine alone does not necessarily tell us what kind of car it is, what does it look like, or how fast can it run. Moreover, because most of these omics technologies are invasive, they require isolated cells and only provide snapshot information about the cell at the time when it is analyzed. Yet in the body, cells can be highly dynamic. To fully understand what a cell is doing, we also need to monitor its phenotype and function over time within their native environment. Single-cell imaging is a powerful tool for this. So far, single-cell imaging relies on fluorescence microscopy. Thanks to a large collection of hundreds of fluorescent dyes and fluorescent proteins, we can now label many different molecules, organelles, and cell types, and monitor their function within living cells or even living animals. However, there are also limitations with fluorescence. You only get to see what you can label. It is challenging to label essential biophysical properties of cells, such as mass, density, and growth. Labeling of small molecules with a dye that's several times larger could potentially perturb their biological function. Quantification of fluorescence can be challenging due to non-uniform labeling, change in quantum efficiency, and photobleaching. Many fluorescent labels require fixed samples and are incompatible with live imaging. For the BIY award-funded project, we aim to develop label-free microscopy tools to image single-cell phenotypes and functions within their native environment. For the past 4 years, we spent significant efforts building new lasers, microscopes, and develop new methods that enable single-cell phenotypic imaging in living cells and animals. One of the main tools we use is stimulated Raman scattering microscopy, or SRS microscopy. Raman is a powerful spectroscopy technique. When a laser is shined on a molecule, it can excite the vibration of the chemical bond and gives off light at a different color. A molecule has many different chemical bonds. The Raman spectrum is like a fingerprint of the molecule. However, Raman is a rather inefficient process. Each spectrum takes over a second to collect. This is where stimulated Raman scattering comes in. By using two pulsed lasers to excite the molecular vibration coherently, it can improve the efficiency by a million-fold, allowing fast imaging of biological samples. How can we use SRS to quantify cell phenotypic properties? Let's start with a simple example. Cells are made of proteins, lipids, nucleic acid, and carbohydrates. They all have different Raman spectra. If we just look at the two dominant species, proteins and lipids, using SRS microscopy, by targeting the two distinct Raman bands, the lipid band and the protein band, we can obtain a two-color image that look like this. The green highlights lipids and the blue highlights proteins. The image may not be very informative at first sight. If we simply convert the color scale to a pink-blue color, we can now see that it resembles H&E staining, the gold standard histopathology staining that has been used for centuries. In fact, pathologists can directly use these images for cancer diagnosis in many different types of cancers with over 90% accuracy without any tissue processing. The simple protein and lipid SRS image contains essential cell shape, size, and tissue architecture information. We can not only image the morphology, but also quantify essential parameters. For example, by performing SRS imaging of protein and water Raman band at the same time, we can convert SRS image into cell mass density with proper calibration. This works not only in single cells, but also in multicellular organoids, despite the ubiquitous night scattering problem. In any optical microscopy, images get dimmer and dimmer as you go deeper and deeper, preventing direct quantification based on signal intensity. Here, because we are using ratiometric imaging of protein to water, the interference from light scattering is removed, thus recovering quantitative density information, which is uniform throughout the spheroid. Applying this simple tool to cultured cells, we can now quantify single cell mass, volume, and density. As you can see here, cell density are much more tightly regulated than mass or volume, which allows us to separate cell types using the density parameter. Moreover, density also changes with cell states. Cells swell by about 10% when they are dividing, and shrink by about 30% when they are undergoing apoptosis. These changes can be sensitively monitored with density. The imaging method is very fast. We can monitor the rapid changes of cell density in response to osmotic challenge at a speed of up to 1 frame per second. You can see the cell respond quickly to osmotic challenge and peaks at about 3 to 4 minutes, and then gradually return back to their original state. Because SRS offers chemical specificity, we can actually take this approach one step further to include not only protein and water, but also lipids. This allows us to quantify both protein and lipid density of the cells in tissue. They exhibit drastically different densities depending on tissue type and cell type. In diseased tissue, such as brain with amyloid aggregations, the hallmark feature of Alzheimer's disease, protein density readily reveals the plaques where you can see high density. Interestingly, we can also see high lipid density surrounding protein plaques. These aberrant features may offer new insights into the origin of plaques. We also developed a new technique to monitor growth of individual cells. This can be done with a method called quantitative phase microscopy. By monitoring the cell mass over time, we can track every single cell in the dish and determine its growth curve. However, it requires continuous mass measurement and can only work for 2D cell culture. We developed a new method to monitor cell growth with deuterium tracing. By culturing cells in medium supplemented with deuterium amino acids, cell growth can incorporate deuterium and allows us to monitor growth rate directly with CD to CH SRS imaging ratio. We have applied this method to monitor the drug response of single cells. Cells responding to drug treatment will have a slower growth rate. This is advantageous compared to the cell proliferation assay. It is much faster and offers single cell resolution. Besides biophysical properties, we're also interested in learning about the metabolism of cells in tissue. One central element of metabolism is oxygen. It has been difficult to measure oxygen in tissue. Oxygen is carried by a protein called hemoglobin in red blood cells. Oxyhemoglobin and deoxyhemoglobin have different absorption features. We developed a technique called transient absorption microscopy to image oxygenation of hemoglobin in red blood cells. Hemoglobin have absorbed one photon to reach excited state, and then absorb a second photon to a higher excited state. By measuring this nonlinear absorption and averaging the excited state lifetime difference between oxy and deoxyhemoglobin, we can measure the hemoglobin distribution and its oxygenation level inside single red blood cells, which will allow us to monitor oxygen delivery through blood vessels. Now that we have these biophysical and functional measurement tools, we can translate them to live animal imaging. We image the live mouse brain through a cranial window. Figure A and B here shows the SRS image at lipid and protein Raman band, like I showed earlier. You can see bright features that are due to transient absorption of red blood cells. We can segment out blood vessels based on these features. The thin strands with bright lipid contrast, those are myelinated axons, so we can also segment those out. The dark spheroid here, those are cell bodies, so we can also segment the cells out. We can image all these features label-free up to a depth of about 300 micrometers. We can then zoom into specific features. For example, The transient absorption signal visualizes the microvasculature. The image shown here is a 3D rendering of the capillary network. The white segment highlights capillaries that do not have flowing red blood cells. Those stored flow can be either transient or permanent. They can lead to local oxygen depletion and has been hypothesized to be the underlying cause of Alzheimer's disease. Here we can see that in mice of all ages, we can observe these features. Those capillaries with stored flow are slightly narrower than flowing capillaries. Moreover, we can further monitor what is causing the stored flow. In many cases, it is the white blood cells that plug up the vessel. We have just begun to apply our oxygen saturation imaging technique to further understand the physiological consequence of stored flow. We're able to see that different capillaries have different oxygenation level. The next step would be to continuously monitor the oxygen saturation change when vessel is stored and how the change in oxygen supply impacts neuronal health. The label-free imaging contrast extends beyond vessels and axons if we leverage the biophysical parameters of cells that can be measured with SRS. Here I'm showing you an example of using machine learning to classify cell types based on SRS imaging features, with fluorescence as the ground truth. Figure A shows the two-color SRS image. B shows the segmented vessels and cell bodies. If we use transgenic mice with endothelial cells labeled by GFP, as shown in figure C here, we can actually train a machine learning model that takes morphological parameters measured with SRS and to predict those cells with 90% accuracy. We can do the same for other vessel-lining cells, such as parasites, with similar accuracy. So here shows an example of the predicted cells for endothelial cells, parasites, and other vessel cells. Once we have the machine learning model, we can apply to SRS images of other mouse brains and start generating a detailed map of structures and cell maps. Here I'm showing an example of all the cells and then classification into endothelial cells, parasites, other vessel cells, and non-vessel cells. So far, we have only used the shape, size, and location information for prediction. In the near future, we will incorporate protein, lipid density, and growth features. We can also further incorporate spectral features. This label-free cell classification approach offers tantalizing hope that we may be able to classify all major cell types in tissue without any fluorescent staining and track cell interaction over time. The machine learning shown here is only one of the tools we can use for prediction. It does not fully use the hyperspectral data sets generated by SRS. We have recently developed a deep learning tool called U within UNET, or UWNet, that offers much better performance in distinguishing subtle spatial and spectral features. For the UWNet, we have a spectral encoding layer nested outside the traditional spatial UNETs. This architecture allows the independent tuning of parameters for image segmentation, classification, and prediction. Here I'm showing an example of predicting live cell organelles with SRS images. The top row is predicted images, and the bottom row is the fluorescent staining after prediction. Cyan color is nucleus, green is mitochondria, and purple is ER. And here shows the three-color overlay from the prediction and the ground truth from staining after our prediction. This powerful algorithm allows us to leverage the spectral information available in SRS and may enable us to predict cell types with much higher accuracy. As a conclusion, we have developed new label-free SRS and transient absorption imaging tools for quantifying single-cell morphological, biophysical, and functional features. We have also shown that machine learning and deep learning enables us to perform label-free cell segmentation, classification, and identification in living animals. For our future work, we hope to build a database of cell phenotypic features for cell classification so that we can trace the interaction of all cells in living organisms under various physiological and pathological conditions. This will allow us to better understand tissue physiology and disease progress. I'm extremely grateful to the generous support of the Arnold and Babel Beckman Foundation. It helped to launch a new direction for my lab that focuses on building instruments for single-cell phenotypic imaging of living biological systems. I deeply appreciate Dr. Beckman's vision in fostering the invention of methods, instruments, and materials that open up new avenues of research and application in the field of chemistry and life sciences. I would like to acknowledge and highlight the group of talented and hardworking students who did all the work. Benjamin Figueroa developed the laser system and the density imaging technique. Fiona Xu worked on the cell growth measurements. Andrew Francis did all the animal imaging work and the machine learning. Bryce Manifold developed the deep learning algorithm. And we are fortunate to receive funding from NSF, NIH, and other sources to continue some of this work. These are the publications funded by the Beckman Young Investigator Award. Please also feel free to email me if you have any questions. Thank you for your attention.